Hi, everyone. In this lecture, we're going to focus on learning about eukaryotic cells. Eukaryotic cells make animal cells, human cells, plant cells, fungus cells. It is not bacteria or archaea. Bacteria and archaea are made up of prokaryotic cells, which were the previous section. So by the end of this section, we'll have a really strong idea of human cells and all these other things. So protozoa are also made up of eukaryotic cells, plant cells, fungus, yeast cells like yeast infections, all made up of eukaryotic cells. And understanding them, again, is very important for understanding diseases, autoimmune diseases, cancer, is to understand eukaryotic cells at a fundamental level. So by the end of this section, you'll be able to identify eukaryotic cells. You'll be able to understand all the organelles and the function of them in eukaryotic cells. And you'll be able to understand something called the endosymbiotic theory, which has to do with cell formation. So starting off with eukaryotic cell structures. So at a big overview, this lecture is going to go over all the components in eukaryotic cells. We're going to talk about the cell plasma membrane, the cell wall, if it is present, cytoplasm with the various organelles like mitochondria, Golgi apparatus, endoplasmic reticulum. So that's what we will focus on. And a lot of times in this lecture, you'll see me say human cells, but just know that I'm referring to eukaryotic cells. And that leads to this slide before we get into the organelles to just briefly talk about some big differences between plant cells and animal cells. So eukaryotic cells, all eukaryotic cells, all plant cells and animal cells are characterized in that they have a nucleus, which prokaryotic cells don't, and they have organelles. But there are brief differences between plant cells and animal cells. One of the main differences is that plant cells have cell walls. Human cells, which are animal cells, do not have cell walls. Plant cells have chloroplasts, which allows them to do something called photosynthesis. And then we as humans cannot do photosynthesis because we don't have chloroplasts. And then finally, some animal cells have flagella, which is used for motility. And they also have lysosomes, which is used to break down old things in cells. And we'll get into the details of them. Okay, starting off with the plasma membrane. So all eukaryotic cells, and as a matter of fact, all cells in general, have a plasma membrane. A plasma membrane is also called a cytoplasmic membrane, is also called a cell membrane. They all mean the same thing. And it's made up of a phospholipid bilayer, just like prokaryotic cells. And it also has proteins embedded in it. Now, one big difference in structure in the cell membrane is that when we look at animal cells, so human cells, they also have something called sterols in their plasma membrane, like cholesterol. And these sterols help maintain the fluidity, which is like the stability of the cell membrane. Cholesterol is so important to maintain like the integrity of the cell because it's in the cell membrane. Without it, our cell membrane would become too fluid, making it unable to regulate the movement of things in and out of cells. And in plants do not have sterols, they don't have cholesterol. Instead, plants have something called the cell wall, which we will talk about later. So here is the cell membrane. Again, it's made up of a phospholipid bilayer and it has proteins in it. The phospholipid bilayers has hydrophilic heads and hydrophobic tails. And the function of it is the main functions is protection. So plasma membranes or cell membranes are there to protect the insides of cells. So acts as a barrier protecting the cell from all the external things it's exposed to. It's also there for sensing the environment. So there's membrane proteins that act as receptors to allow cells to detect signals from their surroundings. It's semi-permeable, meaning it lets some things in and out, not everything. So it selectively chooses what goes in and out. And then finally, one of the main functions of a cell membrane is transport. So the membrane is semi-permeable, it allows selective substances to go in and out, and they can either go in and out either passively, meaning without energy, or actively. We will have a whole other lecture on transport across cell membranes. So unique to eukaryotic cells that we do not see in prokaryotic cells is eukaryotic cells are able to do something called exocytosis and endocytosis, which have to do with the cell membrane rearranging to transport large molecules. So exocytosis is used to get materials out, and endocytosis helps materials into the cell. And we'll talk about it in the transport lecture.
The plasma membrane is selectively permeable. It lets some substances through and not others. The hydrophobic interior of the bilayer is one reason membranes are selectively permeable. Hydrophobic molecules, those that are soluble in lipids, can easily pass through the membrane. In addition, small molecules like O2 can sneak between the phospholipids of the membrane. On the other hand, large hydrophilic molecules like glucose and ions such as sodium ions and hydrogen ions cannot pass through the membrane unaided.
Okay, so we'll have a lecture focusing on passive and active transport after this. Now the cell wall. So some eukaryotic cells have cell walls, but I really want to emphasize that animal cells, meaning human cells, do not have a cell wall. Eukaryotic cell walls are found in plants and algae and fungus, but not in animal cells. And they're made up of carbohydrates, which means sugars. So plant cell walls are made up of cellulose, fungus cell walls are made up of chitin, and yeasts have their own cell walls. So the primary function of the cell wall is to provide structural support and prevent the cell from bursting. So remember that animal cells do not have a cell wall. I'm emphasizing this so much. We human cells do not have a cell wall. And so for our cell support, we have sterols or cholesterol in our membranes. Plant cell membranes do not have cholesterol, and the layer of protection they have is a cell wall. Next is flagella and cilia. Eukaryotic cells may have projections called flagella or cilia, and these are used for movement or moving substances. So they're projections from the cell. For flagella are these long extensions from the cell that help the cell with motility. So for example, sperm cells are an example cell type that have flagella, and sperm cells need to move to fertilize an egg. Not all cells have flagella, but if they do, it's for motility. Cilia are these short little hair-like structures coming out of cells. Not all cells have cilia, but an example of cells that have cilia is some of our respiratory cells have cilia. So for example, in our trachea, the cilia help move mucus and trapped pathogens out of our lungs. So that's why you cough out. So the cilia help trap pathogens so that they don't go deep in your lungs. And a little bit more about flagella here. So flagella is made up of microtubules, and they're made up of tubulin protein. So these structures that help with motility need energy. So they're powered by ATP. And then also, like I said, the cells lining our respiratory tract have cilia on them, and some cells have flagella on them for motility. So this is similar to what I said in the last slide. And defects in this can affect a person with infertility. And also, if your cilia are not functioning properly on your respiratory cells, it may lead to more illness. Now we have the cytoplasm. Cytoplasm just means the inside of the cell. So the cytoplasm is the material inside the cell membrane, but outside the nucleus. So it has the cytosol. The cytosol is the watery part, so the fluid part. It has the cytoskeleton. The cytoskeleton is basically a lot of fibers that provide structural support and help in cell movement. And then we have all the organelles inside, which are suspended in the cytoplasm and responsible for various functions. So the cytoskeleton is very important in maintaining the shape of the cell, especially in moving things inside of a cell and helping the cell move and divide. Ribosomes. Eukaryotic cells also have ribosomes. Ribosomes are where proteins are made. Eukaryotic cells have ADS ribosomes, and they're made up of a 60S large subunit and a 40S small subunit. This is a very different ribosome than the ribosome found in bacteria or in prokaryotic cells. Prokaryotic cells have a 70S ribosome. Eukaryotic cells, human cells, have an 80S ribosome. Ribosomes are either membrane bound, meaning they're attached to the endoplasmic reticulum, where they make proteins that are exported from the cell or inserted in the cell membrane, or ribosomes can be free, found floating in the cytoplasm, where they make proteins that function inside the cell. And this is a very important slide, especially when we think of bacterial infections and microbiology, because, for example, we have some antibiotics that target the 50S ribosome. So if I ask you on an exam, if an antibiotic targets the 50S ribosome, what effect would it have? You would say it would affect bacteria, because bacteria are prokaryotes. They have a 50S ribosome, but they would not affect human cells because human cells do not have a 50S ribosome. Next, we have the nucleus. So in eukaryotic cells, a defining feature is that they have a nucleus. The nucleus, we say, is a control center of the eukaryotic cells, and that's because it houses the DNA. That's basically the function of a nucleus in eukaryotes, is to protect or house the DNA. The nucleus is surrounded by something called the nuclear envelope, and the nuclear envelope is just a double membrane that protects the DNA in the nucleus. There's also something called the nucleolus, and this is where ribosomes are made. And then there's also nuclear pores, which are these openings or holes that allow material to move in and out of the nucleus, especially when we think of wanting to make a copy of DNA when DNA is in the nucleus. You may want enzymes to go in and out. So nuclear pores are important, and they're also important for exporting other things. So again, the main function of the nucleus in eukaryotes is to house chromosomes or DNA. DNA and chromosomes mean the same thing. So if you say you're looking at someone's chromosomes, it means you're looking at their DNA. So when we think of eukaryotes, in the nucleus, DNA exists in two forms. Chromatin is the relaxed form of DNA when the cell is not dividing, so that the cell can make copies of the DNA. Chromosomes are during cell division when chromatin condenses to form these visible chromosomes. And this makes sure that genetic information is accurately passed on. And so basically, to summarize, chromatin, chromosomes, DNA all mean the same thing. They're just a form of DNA. So in short, chromatin is the loose form of DNA, and chromosomes are the condensed form that's used during cell division. And again, DNA is in the nucleus in a eukaryotic cell. Next, we have mitochondria. Mitochondria are the powerhouses of the cell. That's just a fancy way of saying that's where energy is made. So the reason we have energy is because of the mitochondria inside of our cells. The energy molecule of cells is ATP. So inside of your cells, you have mitochondria. Mitochondria make ATP molecules. ATP molecules is energy. So if you can figure out a way to create more ATP in someone, you'll be able to create like an energy drug or something like that. So when we think of the mitochondria also, there's two things that are unique about them, is they have a single circular piece of DNA of their own, and they have their own 70S ribosome. The reason I say this is important is this led us to the theory that because mitochondria have 70S ribosomes and their DNA, maybe they have a bacterial ancestor, which we'll talk about later in the endosymbiotics theory. But the take-home message from this is the function of mitochondria is to make energy, meaning make ATP. Now, the structure of mitochondria is important. So mitochondria have a double membrane structure. And so this is important because the mitochondria with this double membrane structure, There's a lot of these folds called cristae. I'm going to simplify this as much as possible. And this is where you form a lot of the energy or the ATP. So the more cristae you have, the more surface area you have to make ATP. And when we learn about cell respiration, we'll learn about that. Next, we have chloroplasts. Chloroplasts are present in plant cells, human cells, animal cells. We do not have chloroplasts. Chloroplasts are where photosynthesis happens in plant and algae cells. So, like mitochondria, they also have their own DNA and 70S ribosomes supporting the theory that there is a bacterial ancestor. We'll talk about that. So chloroplasts are very important for plant cells because they convert light energy, sunshine, into chemical energy, meaning plants can produce their own food. That's photosynthesis. Next, we have the endoplasmic reticulum, also called the ER. The ER is basically a bunch of flattened sacs, and it's responsible for producing, processing, and transporting various things for the cell, like protein. So we call it the factory of the cell. It's where things are made. So there is a lot of protein made on here. And remember, I told you that the thing that makes protein is ribosomes. So when we think of the ER, if I tell you that the ER makes proteins, there must be ribosomes involved. So there's two types of ER. There's rough ER and smooth ER. Rough ER is ER, endoplasmic reticulum with ribosomes on it. So that's where proteins get made. Smooth ER doesn't have ribosomes, and that's where lipids get made. So that's what ER does. It plays an important role for making molecules for the cell, like proteins, like lipids. So again, rough ER has ribosomes on it. That's why it's called rough. And the function of a ribosome is to make protein. So this is the area that receives, transports, stores, and makes protein. And then smooth ER does not have ribosomes on it. And it's usually there to make things like fats. Lipids means fats. It helps make cell membranes, hormones, various other things. Next, we have the Golgi apparatus or the Golgi complex. The Golgi complex is basically the area of the cell that tells things where to go. So we say it's the post office of the cell. It processes, sorts, and ships proteins and lipids that were made in the ER. So proteins are modified in the Golgi complex where they're folded, tagged, and packaged into these things called vesicles so that they can be transported to their proper destination. Either that's inside the cell, on the cell membrane, or outside the cell. So this organelle is very important for maintaining cell structure by telling things where to go. That's the Golgi apparatus. It assembles raw materials and routes them. So vesicle transport. So in cells, there are these things called vesicles. They're basically these small membrane bound sacs, and they transport materials within the cell. They play an important role in moving proteins and lipids from the ER. Remember, the ER is the factory of the cell to the Golgi apparatus. Golgi tags them to go for wherever they need to go, and then into a vesicle. So maybe if you have like a membrane bound protein that needs to be made, it will be made on the rough ER, and then it will go to the Golgi complex where it will get tagged to go to the membrane. It will be put in a vesicle and it will be told to go to the cell membrane. Lysosomes. Lysosomes are, we say that they're the recycling center or the trash can of cells. So these are membrane bound vesicles that have these enzymes that can break down waste materials like old organelles or various debris in the cell. So in immune cells like phagocytes, there is lysosomes that help break down pathogens by eating them up. So lysosomes are these important vesicles that have digestive enzymes and they destroy harmful bacteria, they destroy old organelles. And again, phagocytes are white blood cells in our immune system that try to break down pathogens. So they have a lot of lysosomes in there. And just as an example, there's something called lysosomal dysfunction, and this is a disease where The lysosome is not functioning properly, and so waste products accumulate inside cells, and this leads to cell damage. Okay, next, so more about the lysosome. Here's the lysosome. Here's a vesicle that has a damaged organelle. They fuse together. The enzymes will break this old mitochondria, and then these components can be used to make other organelles. And then finally, I want to talk about with the endosymbiotic theory. So there's something in science called the endosymbiotic theory, and it suggests that eukaryotic cells, like human cells, animal cells, plant cells, evolved from prokaryotic ancestors like bacteria. And the reason why this theory is supported is because eukaryotic cells, for example, plant cells, have mitochondria and chloroplasts. If we just think of mitochondria, mitochondria have their own DNA, which is circular, similar to bacteria, and they have their own 70S ribosome, which is similar to those found in bacteria. So this theory tries to explain how eukaryotic cells acquired these organelles. Basically, two prokaryotes somehow ate each other, and that ended up being a eukaryotic cell. And now to just summarize what we've covered about eukaryotic cells, they're larger and more complex than prokaryotic cells. They have a nucleus, which prokaryotic cells don't. The nucleus houses the DNA, and they have organelles like mitochondria, which makes energy. Endoplasmic reticulum is kind of like the factory of the cell. Golgi apparatus tells things where they go. Lysosomes break down waste materials. So that is eukaryotic cells. So at the end of this lecture, I'm hoping that you are able to answer these questions. So these are important questions to think about. So if we were to list various eukaryotic cell structures and go over them, we could talk about things like flagella. Flagella is important in motility. We could think of things like the plasma membrane. The plasma membrane is important in transport of substances in and out of a cell. Actually, let's go over a few that are important that I hope you got from this. So again, there's a nucleus. The nucleus contains the cell's DNA. There's the mitochondria, which is the powerhouse of the cell that generates ATP. There's the endoplasmic reticulum, and there's two types. Rough ER has ribosomes on it. Ribosomes make protein, so protein is made on rough ER. Smooth ER doesn't have ribosomes, and it's involved in making lipids, which are fats. Then we have Golgi apparatus, which modifies, sorts, and packages proteins and lipids for transport. We have lysosomes, which contain digestive enzymes that break down waste materials in cells. Remember, prokaryotes don't have lysosomes. We have vesicles that can hold things for transport. We have flagella, which are important for motility. We have cilia. Cilia were not seen in prokaryotic cells. They're short hair-like projections that can move substances across the cell. For example, there's cilia lining our respiratory cells. And then there's the cytoskeleton, which is protein filaments like microtubules, microfilaments, intermediate filaments that give the cell structure and hold organelles. And then we describe the structure and function of flagella, cilia, and cytoplasm. So again, eukaryotic flagella is made up of microtubules. It's powered by ATP. Cilia are shorter than flagella, and they help move things like the respiratory tract. The cytoplasm is the inside of the cell, and it contains the cytosol, which is the watery part, and the organelles. Next, we compare the structure and function of eukaryotic and prokaryotic ribosomes. Eukaryotic ribosomes are larger. There's 80S ribosomes. They have a 60S large subunit and a 40S small subunit. Prokaryotic ribosomes are 70S ribosomes. They have a 50S ribosome and a 30S ribosome, and they're targeted by certain antibiotics because their structure is different from eukaryotic ribosomes. So for example, there's an antibiotic called erythromycin, and it binds to the 50S subunit of bacterial ribosomes. So it stops protein being made in prokaryotes without affecting eukaryotic cells, because eukaryotic cells, human cells, we do not have a 50S subunit because eukaryotic ribosomes have an 80S ribosome. So the question, I love this question. The antibiotic erythromycin binds with the 50S portion of a ribosome. What effect does it have on a prokaryotic cell or a eukaryotic cell? So because it binds to the 50S subunit, it affects prokaryotic ribosomes and it stops protein synthesis. It does not affect eukaryotic cells because eukaryotic cells do not have a 50S portion. They have an 80S ribosome, and in the 80S ribosome, there's a 60S large subunit, not 50S. Next one is describe the function of the nucleus, ER, Golgi. So we went over that. We just went over all of those. Compare the structure of the nucleus of a eukaryote and the nucleoid. Prokaryotes do not have a nucleus. We just call the area where the DNA is found the nucleoid. Eukaryotes do have a nucleus. It's a membrane-bound double membrane that holds the DNA. And then the difference between rough and smooth ER, rough ER has ribosomes. That's why it's rough. It makes protein. Smooth ER doesn't have ribosomes and it's involved in making fats or lipids. And then finally discuss evidence that supports the endosymbiotic theory. So this theory says that maybe eukaryotic cells evolved from a prokaryotic ancestor. And the evidence is that mitochondria and chloroplasts both have their own DNA. And also they both have 70S ribosomes similar to prokaryotes. So this evidence suggests that mitochondria and chloroplasts originated from prokaryotes like bacteria or archaea that were eaten up by earlier cells, and this forms a symbiotic relationship. So that is our lecture on eukaryotic cells.